So let me start. I hope that now it works. Yeah, so I just wanted to sort of give a motivation for this work and the presentation. And I just wanted to say that this is not a classical talk where we would like to do some kind of data transmission. We want to study, for example, how source could be encoded and decoded maybe, and also some kind of coding for the channel. We are not interested when it comes to the transceiver design, how the transmitter receiver systems can be designed. And also we are not interested when it comes to some of those performance metrics that we normally use, like, quality of services like channel capacity, bit error rate, and so forth. So that's kind of the background information I would like to sort of say that, this is not a classical talk, but this talk is slightly different. And I would like to sort of emphasize that the reason for what we would like to do is we are interested, since I'm working in a research center, clinical research center in Oslo, as well as a professor in the university. So I'm mainly interested in cell-to-cell communications. And for reasons is that I would like to understand how this is done, for example, in human body, and whether we can learn from the nature, and then how we can use our knowledge to create models mimicking the biophysical mechanism that is taking place, and then to see whether that could be useful to develop new applications. So theranostic, I don't know whether you guys have known this term, is meaning like it could be a therapy or diagnostic, or sometimes it could be as a joint application, like therapy diagnostic in the same application. So in our body, it's containing so many different parts, so like organs, vascular systems, nervous systems, and so forth. But the basic building body, that is actually a cell. So cell-to-cell communication seems to be very important to understand, and it's a big area, and it's considering from cell biology all the way to the molecular biology. So this is the field that comes from biology. So why electrical engineer like me is interested is to see whether what I can do to contribute to their field, and also creating new application in medicine for us. Hopefully that could be useful to create new therapeutic solutions for different diseases. So in electrical engineering combined with biology, there is a field called system biology. System biology is a field that's been there for the last 40 to 50 years. And they study, for example, the observations on a cell level, or experiments that could be represented like differential equations. But they don't study what is happening, those changes that is taking place, for example, in cell 2, and how these changes are taking place, what is happening on cell 1, where there is a channel is in between them. And maybe that's the way that engineers like us coming from communications potentially can provide other methods and tools to understand this part and creating this into a communication system. So my talk is going to be in this way, and I would like to sort of start with cell, and then I will go a little bit towards in this channel part and the receiving cell, and then I will explain this my research using some examples. So as I mentioned to you, the benefit of this work potentially can lead to complement the knowledge from system biology or computational biology with our knowledge coming from our field. And the other part that I think it would be also quite interesting to explain is the spatial temporal granularity that we can provide. Because when we do in vitro experiments or in vivo experiments, the time points between two observations can be quite long, maybe hours. But we can provide, using our model, both space and time, that they can provide interesting information. So if you talk to a biologist, he will be very much interested about the steady state system. So, when after some time everything converges, and he is interested only what is happening in 6 hours or 24 hours or 48 hours. He's not so much interested about intermediate dynamics that is taking place. But that might provide very interesting information when it comes to different type of disease situations going from health to disease. And this is actually a new area that people have started looking into that. And we engineers, I think we can also provide interesting tools to that. So the other part actually is to reduce the experimental cost using our models. Potentially also can develop into new tools when it comes to diagnosis, and also maybe deliver drug delivery systems. So the image that you see, the figure on the right side, in a normal cell, a biological cell, an artificial cell could be developed. So when it comes to human applications, so we cannot just insert any artificial cell that we can create in the lab. So normally what we do is we take the human cells and then we engineer them, and then we can insert them inside again and using something called stem cell biology. So I will not go into all these biological terms and different methods. I just wanted to show that it is still possible, that is actually used today in clinical medicine. So let me come to the topic today that I just wanted to mention that I am interested, my group is mainly interested in something called extracellular vesicles. And extracellular vesicles, as you see it on the right side here, the cell. And then in the cell, you find many parts. And we are mainly interested into something called vesicles. And it could be mentioned as exosomes. These are like small vesicles. I will come back to the size and everything in a minute. But it also exhibits so many different types of vesicles that you see it here. These vesicles are used, for example, when it comes to cell-to-cell communication. On the left figure, you see, for example, something very close. The two cells are, very close to each other. They could be also connected by something called gap junction. And then the communication between these cells are by the means of chemical signaling. So they are basically chemical signaling. But also long distances, for example, if it's going to be through our circulatory system, far away cells could be, can be send messages to cells from a distant area, and that could be using chemical signals also. And this potentially is called as endocrine. And then we have something on nervous systems. And as you see here, a neuron, for example, connected by an axon, and is trying to sort of convey the message to a distant signal, a cell, then it's called as a neuroendocrine. And I'm not going to go through every of them, but I will just focus on something on local cells are not very far away, but it's going to be conveying the messages. And we will explain to you using two different types of environment, one for the brain and one for the heart. So for example, here, then I'm interested to show you this figure showing that, we can relate this work as communication engineers or wireless communication people, that there's something called transmitting cells, and they release certain molecules. So here, for example, what we call it as extracellular vesicle. I introduced you a little bit about that. The extracellular vesicles, as you see it here, the smaller one is called an exosome, and then we have microvesicles, and you see the cell here. So these are like small ones from 30 to 100 nanometers. The interesting properties of them is that they can carry some genetic material. So they can carry a cargo within themselves. So the cell releases them, and they can also engineer to carry certain information. The other part actually is that these are so small that they can penetrate some biological material barriers, like blood-brain barrier. So if you want to signal to deeper part inside the brain through the cardiovascular network, you might be able to use exosomes. And the other part actually is that interesting is that exosomes cannot be destroyed by immune system. So we have interesting properties that is exhibiting extracellular vesicles that is actually interesting for us to study whether we can use them both for signaling to the distant cells through the cardiovascular network and also to deliver some cargos, for example, as a drug delivery solution. So just to give you an example of these works, actually started by us and then went into this EU proposal and got accepted as an EU FetOpen project. It's called GLADIATOR. And here, for example, the idea is to use this concept to treat, for example, a brain cancer situation called glioblastoma. The glioblastoma cancer, unfortunately, cannot be cured with the current methods just because there is no such treatment available. And the current nanoparticles that people are using, unfortunately, they don't penetrate deep inside through the blood-brain barrier. So you need to find a different solution. This is actually a completely cell-based, so it means that it is biocompatible method using the human cells and then using an external signal to control the release of these vesicles. Hopefully that goes to the target location. So that's another set of cells. Here, for example, is the cancer cells. And then we would like to understand what happened to those cancer cells. Are we able to deliver our vesicles to them and download the content, the genetic material, so that we are able to reprogram the cancer cells and, for example, kill them? And then we would like to understand what is happening to that in a closed loop situation, because sometimes we inject certain things and we hope that this is going to work. And unfortunately, most 90-95% of the time, they don't work. So we would like to understand whether we can have a receiver system so that we can control this in a controlled manner, like a feedback loop. Let me not go too much into the details here, just to give you an understanding how we are doing in this project. Is we use something called radio frequency signals from external source. So outside the brain, outside the skull of the brain, and then we can excite the implantable cells. It could be releasing these exosomes. The other method I mentioned, you could also make them externally. Then you can inject them through the cardiovascular network, and then we would like to see whether they go to the target location, and then we would like to understand what is happening to them. So here in this application, you see that it has both a drug delivery solution. It also looking into something reporting back from those cells. These are again these blue dots that you see here. They are basically the extracellular vesicles. So extracellular vesicles, one part is delivering the drug, the other part is released by the cancer cells and be used as a diagnostic information to see how effective the therapy is going on. Then we can titrate the therapy by using an external signal like RF here. So this is for the brain. And then I would like to show you another application that we have. It's funded by the Research Council of Norway. And then this is for the heart. The same idea here using extracellular vesicles, but this is actually a heart condition called heart failure. So these are patients who we consider them as a terminal patients. Either they need to have a new heart or heart transplantation, waiting to get a new heart, or we may have to use a mechanical heart, like a pump that we can, insert and use it as a intermediate therapy until they get a new heart through a heart transplantation. So the reason for them is that the heart muscle, as you see here, it has become quite stiff. So heart, as you know, that is basically a pumping machine. It collects the blood from the rest of the body, sends to the lungs, get oxygenated, and then pumps again to the rest of the body. When the muscles are becoming stiff, it is unable to pump. It's unable to contract properly, and this stiffness is quite interesting. And I will show you some example of how we study this part, because we are interested to see whether we can use extracellular vesicles as again a therapeutic tool or therapeutic means to treat this disease. And I will not go into all the disease situation and how this is being done, but this is actually an overview of this project where you can use, for example, optical signals. Then if you want to use an optical signals, you will have to go through that using something called nanoparticles. The RF part that we have been using in the brain project, we can also use it here. And also we can also use ultrasound. So ultrasound external signal to control the release, and then to see what is happening to the target cells and get diagnostic information in a closed loop manner to treat heart diseases. So let me come back to the idea about EV as a transmitter. Now we are on transmitting cells, style number 1, if you remember one of my earlier figure. And here I would like to explain that how this can be done. So cells normally release EVs that can be used as a diagnostic information. Why? If the cell is malfunctioning, then EV will carry that information. So looking at the EV content, we can say that the cell is healthy or a disease. Okay, so that would be a diagnostic information. The other part actually is that understanding where those cells are, then we can also understand where the problem lies, disease location and the status information. And then we can use this information also in the feedback loop. That is actually when you release the EVs from cells and engineer them to carry certain genetic content, like DNA or microRNA, and they go to the target location. And then if they deliver that, then it's considered as a drug delivery solution. And then whatever happens to that cell could be used as a feedback loop, because they also release another set of EVs. And that can be used as a diagnostic information. Okay, so the cells can be used in this way. So from a transmitter cell point of view, let me explain you a little bit about what is happening within the cell. So in the cell, the transport mechanism is something called Golgi apparatus, and this is actually encapsulating these particles or the EVs into something called multivesicular body, MVB. And then the MVB goes to so this is everything is happening inside the cell, and then it goes to the membrane, and then it's fused to the membrane, cell membrane, and the particles are released. So EVs, as you see it on the right figure here, the EVs are released by these multivesicle bodies when they are fused with the cell membrane. But we also have some way of regulating this part using these different channels as you see here. Calcium plays a very important role. We also have potassium and sodium. And also I mentioned to you that we can use external signal, for example, like RF or ultrasound, optical signals, or we can even inject direct current to them. So some of them are excitable, like electrically excitable cells, like neurons, cardiomyocytes, these are heart cells, alpha and beta cells. Those are like associated when it comes to diabetes. So let me explain this part. When it comes to the building blocks of EV release part. So the first part is about that this has been this equation has been actually proposed by Hodgkin-Huxley's model, and if you know that these are the guys who got the Nobel Prize in 1963 for this work. And this is, as you see it, that the membrane, the cell membrane voltage can be a function of voltage gated potassium, sodium, and the input current. So that is actually what you could sort of use as a controlling way of the membrane of the cell. Then we have the other parts. So we have the calcium, the L-type calcium channels that could be explained in this equation as you see it here, the equation is one here. And the calcium current go through the calcium voltage, voltage gated calcium channels. You see it here. And there is something called submembrane. And we would like to, as we are interested about the EV release rate, so this is associated, the first is associated to the L-type calcium channel. The second part is associated with the submembrane of the calcium. So you see these two parts associated with the membrane, and we can go all the way back to the control current, for example, how we can control the release rate. Let me show you some results here. So when you have the control signal, so these are the currents. So you need to look at the left figure, depolarization part. I believe that you can see my cursor. And then if you increase the current, the control current, you see that also the activation. So the cell becomes active, depolarization is taking place, and it's firing. When it is firing, the bottom figure here, it could be associated to the calcium level channels. The calcium levels are shown in the middle figure here. For example, when the current is the purple one, the 20 microampere current that you see here, then the purple curves are associated with that. So this is actually the calcium levels. When the calcium levels are increased, then you also see that increased levels of release rate. So the purple one, as you see here, then we have 20 ampere input current. When the current is 5, the blue curve here, then you see that it's a little bit less. So you can use the input current to sort of regulate this part. So this is actually a direct regulation of the cell, and we can control the release. Then you need to have an electrode or a microelectrode to do that part. But can we do that with an external signal? That was actually our interest. So you have an implant, then you put an external RF, for example, antenna outside on the skull, and then try to see whether you can excite the cell from a distance. And this is an experiment that we have done in collaboration in the project with a group from Finland. And we have developed an antenna system that we have integrated in the cell culture model, as you see it here. It's a cell 6 well plates, and then we excite those cells, as you see it here, and then those cells also have these extra vesicles there. It's a group of cells. And then we have used, for example, 2.4 gigahertz and also 433 megahertz. So 433 megahertz actually is associated with something called mixed band, medical implant communication system band. So that is actually regulated and can be used for implant communications. So you can excite an implant and using that frequency. That is allowed by the standard. So we would like to study, and we have some interesting results. We just don't want to cook the cell. RF could be considered as, can increase the temperature. And then, of course, temperature elevation also can lead into release of extracellular vesicles, which is not our interest here. So this is actually the work that we have done. Now I see that my time is also running. So then we have, as you see the figures here, the nanoparticle tracker can give information about the number of particles that we have. The fluorescence imaging that we have done is also give how these vesicles associated to those cells, how they are moving away from those cells to the target location. And this figure on the right side is basically the results. And then you see, for example, with and without degradation, it means that some of the cells can basically, degrade and vanish, or can be taken up by other cells. So this is actually the channel. So I think I need to go a little bit faster. So the channel's in between here. And then I would like to sort of explain the channel, which could be a model using total city volume fraction and non-isotropic microenvironment. And this is basically reasonably well known. But the issue that I mentioned about the half-life and some of the extracellular vesicles that don't bind to the target location, we need to take that into consideration. And there is some other interesting properties that we have also taken into consideration. It's not only the ligand binding that most people do, but we have something called clathrin-mediated endocytosis. It's the receiver process that we have to also consider when it comes to the channel part. And then this has been, modeled using this finite element method. And this work was done by Hamid, the PhD student. And this figure that you see here is based for the heart. As you see, the heart rate, 120 beats per minute. And the EV release rate in the blue curve here, the control signals, that's basically the injected current. And the EV distribution is shown in this figure that, the video that is played here. So as I mentioned to you, just because we are interested about heart failure situation, so we would like to understand this cardiac extracellular vesicle space in the cardiac extracellular matrix. And then it contains both soft tissue and stiff tissues, like, the stiffness of the heart, just because I mentioned that it may not be pumping very well just because it's so stiff, the muscles are stiff, and we would like to understand. And then if you see, for example, if you use a normal diffusion type of situation, that phi is equal to 1, and then you see this curve here, which gives you a high concentration. But unfortunately, that is not true, because we cannot receive high concentration when there is a stiffness involved in this part. So when we increase going from soft to stiff tissues, or the elasticity is reduced, then the concentration levels, the receipt concentration levels also goes down. So these are the analytical solutions that has been done by Hamid, where Martin Damrath has developed a particle-based stimulator. And then, we show both analytical solution and the particle-based simulation results. And there's a good correspondence between these two. So I think I will just jump over this part and then go to the receiving part of the EV as a receiver. As I mentioned to you that, we are interested that the EV receiver can be considered as a biosensor. So there is a EV could be received by normal healthy cells. And if they are coming from sick cells, cancer cells, for example, that could be used as a biomarker to titrate the therapy. So transmitter channel and the receiver part. So when it comes to the receiver, basically it is a little bit more complicated than just looking into ligand binding. Most people normally do that. Here we have many uptake mechanisms that are deployed at the same time simultaneously. So as I mentioned to you, the cadherin-mediated endocytosis, ligand binding properties, fusion, and so forth. So I don't have the time to go into all the details, but here is an interesting work done by Miladin. He was trying to do this in the nonlinear system that is about the cadherin-mediated endocytosis, that's the receiver process. And then he was using the Volterra series and the expansions and the kernels there, and making into a linear model. And having this linear model shows that we can find the input-output relationship, like a transfer function or the impulse response. And then he shows that the differential equations results are here. Then if we add, for example, 3 kernels, then it comes very closer to that. So that's one way. The other one that it was mentioned in one of those slides is about the fusion. And now this is not our work, so I will not spend too much time, just to say that there are other groups that have started looking into that, which is very good. And then I just wanted to show you some results when it comes to the biological cell as a receiver. So these are the experiments that have been done by our collaborator, Masamitsu Canada from Michigan State University. And here you can see that the results that we have from other works that we have done with Mohammed, and the correspondence between the experiments and the model, the numerical results. So just to summarize that, we were able to know a little bit more about how cell-to-cell communication is working when it comes to brain and heart applications. And how this could be explained both when it comes to in vitro experiments and new concept when it comes to delivering drugs, and both for the heart and the brain. And I just wanted to thank my group, especially Miladin, as you see it here, and Martin and Damrath, Mohammed Sufahari, and Hamid Rasari. These are the main people who have contributed to this talk today, and I would like to also thank our collaborators, Chris Contact from Michigan State, where Hamid is doing some experiments there now, and Seppo Vaininen from Finland, and Bill Luch from Australia. I will also thank our sponsors for this research Council, European Union, and Health Southeast and Innovation Norway. And thank you very much.